It's a cue to trigger a memory, but then maybe it activates feeling the riding on the camel experience and some knowledge about camels and that it was a one-hump camel and so on. So these things are highly interactive and hierarchical. There's the hippocampus. It's not actually blue in your brain. And the surrounding structures in the medial temporal lobe, medial being in here, temporal being this bit. So that sort of set of structures is considered to be at the root of the core of the episodic memory system. I will also lapse into saying autobiographical memory system, and I mean that pretty much the same thing in this talk if I forget to be consistent. Hippocampus receives input via indirect levels of processing from actually not just the two prominent visual streams, dorsal and ventral, but multiple association areas and emotion from the amygdala and other structures associated with emotion. And even Fellman and Van Essen's circuit of the primate visual areas, they put the hippocampus at the top of that hierarchy, interestingly. And it makes sense. It does receive convergent input from all of these areas. So that's a sort of quick look through the medial temporal lobe. And another term you're going to hear a lot in this talk is the concept of the default mode network. The default mode network was thusly named originally because it was thought of as the brain's idling mode or resting state mode. Sometimes people still call it the task neutral network. And I very vigorously object to that term, as you might have guessed, because this network is also engaged in tasks. It's not just task negative or task neutral. Episodic memory retrieval, future thinking, spatial memory retrieval, spatial navigation and planning, and theory of mind, to name a few tasks where you engage the default mode network. What is probably fair to generalize about it is that it's more self-reflective kind of processing, internally driven, as opposed to externally cued, being on a cognitive task and focusing with attention that requires executive functions. Okay, so that's the background. Let's talk a bit about spatial memory in particular. And this is going to come back, so think back on what I say about visual transformations and the transformation network when we get to talking about trauma memory a little bit later in the talk. So my former PhD student, Pat Byrne, did this very nice work where he developed this super complicated model. It's been nicknamed the BBB model because it's by Byrne, Becker, and Burgess. And I am not going to go into any of the details, but feel free to ask or look up the paper. But just suffice it to say that at the core of the model, a number of different modules, the hippocampus / medial temporal lobe was thought to represent a spatial map like this depiction aerial view of the Milan Square and the cathedral in an allocentric or world-centered framework. Whereas the posterior parietal regions were thought to represent things in the world in egocentric frames of reference and to subserve things like mental imagery. And so if you want to form a map of a new place, your visual system delivers you the information in this form. You need to somehow map it into this form. How do you do that? There must be some kind of coordinate transform. It might not be literally going from x, y coordinates, x, y, z coordinates in one frame to another, but somehow the representation must get transformed into allocentric framework. And based on quite a bit of neuroimaging evidence, we postulated that this area in between that interconnects both of these regions, the retrosplenial cortex, it's in the posterior cingulate, which wraps around the midbrain, is the site of the coordinate transforms. And then the prefrontal cortex also needs to come in when it comes to maintaining information in working memory. So when you recall a place or any episodic scene or event, you might initially use all this machinery in the medial temporal lobe to recall the event, but then if you're trying to maintain it in memory, you're going to use these executive control centers, prefrontal regions, to keep that active in working memory. So that was later brought in by Pat in the subsequent paper as part of this model. So I won't, again, go into model details, but I will just briefly tell you about one really neat fMRI study that another PhD student, Corrette, ran, where he actually put people in the scanner, had them navigate around a virtual scene. This is an aerial view. It's an arena. And this is what view they would actually see. There's an avatar looking at an object, and there's a number of distal landmarks visible. And what Corrette did was have people go around and learn the locations of objects by navigating and performing a collect and replace task, bumping into the objects and then replacing them later. And then they were tested for their memory for the object locations from different viewpoints. And either the same viewpoint as during study, or from a rotated viewpoint, or from a translated and rotated viewpoint. And the third one was the most challenging. People refer to that as the judgment of relative direction. Imagine, this is the cue literally they saw in the scanner. Imagine you're standing at the red square, facing the pink circle, now point to where the blue triangle would be. So you need to recall the whole scene. If you are good at allocentric remembering, you would recall those landmarks and situate yourself within the scene in the right viewpoint, and that would require, that would heavily tax your viewpoint transformation network. And just to cut to the chase, when people were engaged in that most challenging JRD task, they most strongly activated this network of regions, the precuneus in the posterior parietal cortex, retrosplenial cortex that we thought would be important for perspective transformations in the posterior cingulate, and hippocampal and parietal regions. So it looks like they were using that whole circuit, and moreover, it wasn't just active, but the activation statistically correlated highly with performance. So that's a really nice validation that, yeah, the activation wasn't just sort of incidental, but it actually correlated with the ability to perform the task accurately. All right, so if you don't remember anything else, remember retrosplenial cortex in the posterior cingulate. That'll come back to haunt us a little later. Okay, so I want to now broaden. I started at the hippocampus. I broadened out to the default mode network and the medial temporal lobe. And now I'm going to talk about multiple networks. The default mode network being one, and two others that my former PhD student Saurabh studied. And Saurabh has continued to collaborate with both Selena and Mohammed and other students in the lab. He's quite a powerhouse guy. I do recommend him if you can get your hands on him. Yeah, so we started with this model, this triple network model proposed by Menon, that proposes that when you are, you're either predominantly engaging your default mode network or your central executive network, whether you're doing sort of self-referential cognitive tasks or externally driven cognitive tasks. For instance, in the study I'm about to describe to you, we set up alternating memory tasks that would switch at random between autobiographical memory retrieval and working memory, a two-back task. And this other network, the anterior salience network, is postulated to help you in task switching between the other networks. So you would expect when you're doing, say, episodic memory tasks, these two would couple strongly together. And when you're engaging in working memory, these two should couple strongly together. So what we did initially was simultaneously collect EEG and fMRI while people are performing this task, alternating blocks of autobiographical memory retrieval and working memory. So before they went in the scanner, they generated narratives of some autobiographical memories. and they self-identified keywords that would be good for retrieving the memories, like oven, icing, cake decorating, whatever, right? So there would be some memory about making a cake. These would be keywords that could later be used as cues in the scanner. The working memory task would just have, like this task, they'd be just reading a sequence of words, but they would be asked to occasionally perform a two-back. Which of these 4 words did you see 2 words ago? In the autobiographical memory or episodic memory task, it was, here are 4 words, which of them was part of the memory you were just retrieving? Sort of as a manipulation check to make sure they're on task. The critical part was scanning them while they're either retrieving these kinds of memories or performing these tasks. And the other thing we expected, besides that this would activate the default mode network and working memory would activate the executive network, was that at the time, unpredictably, when they were asked to do a switch from autobiographical to working memory, we would expect the salience network to kick in and then maybe also couple more strongly with the task-appropriate network, whichever it was. So that's the setup. What we really wanted to do, though, was free ourselves from needing to use fMRI to track these networks and see if we could develop features from the EEG alone that could subsequently be used. No one had really done this before. People had come up with some tentative EEG markers of default mode activity and so on, but no one had really come up with a set of features that would allow you to accurately classify which network was being activated. So this was Saurabh's pretty fabulous PhD work. And this is just an example trial, single trial from one participant showing from the fMRI, initially we're looking just at the fMRI, how the default mode network structures would be nicely activated during the autobiographical or episodic memory trial, and then they'd taper off and the central executive would ramp up during the working memory. Interestingly though, if you look at a group average, during autobiographical memory, initially there seemed to be a retrieval bit, but then the central executive would tend to kick in on the later part of the trial. And that makes sense when you think about it. We hadn't predicted it, but we might have if we looked more at the literature. It makes sense that you're in more memory maintenance mode for the later part. Once you've retrieved the memory, you're trying to keep it active. So that was also kind of a neat result. And then the salience network shown in green is nicely co-activating here with, sorry, I said this backwards. Just reverse everything I just said, okay? This is the autobiographical memory onset, so the default mode network is kicking in, yes. And the salience network nicely co-activating. Earlier it was, and there's the late onset of the executive function network. Yeah, sorry about that. So that was nice and validation just from the fMRI. Now what did he do with the EEG? This gets complicated. I'm not going to go into these details. You can obviously all read that and follow all the details, so I don't need to explain it. But just in short, he applied a feature extraction pipeline to the EEG and a classifier at the bottom. And the way he generated the labels for the classification was from the fMRI. So he's sort of using the fMRI data stream to teach the EEG data stream how to recognize, based on whatever features it could, those 3 networks. So now we have this set of EEG-only features. And this just shows projecting them down into a 3-dimensional space. They do separate very nicely, the 3 networks. And so next, okay, that's a preliminary conclusion. We have those EEG features. Now, can we test this by trying to push those networks around? How can we push the networks around? Well, one way is to use a multi-week intervention that we think might modulate these networks and see what happens. So the next part of my talk is doing just that, trying to test this set of EEG features to see will they actually track as expected under lifestyle interventions. What lifestyle interventions, you might ask? Well, aerobic exercise and neurofeedback. How many of you know about the Muse headband, the Interaxon Muse? Some people have heard of it. Well, it's a commercial grade 4-electrode headband, and they sell it as a mindfulness app. It's neurofeedback, which is meant to emulate some degree of mindfulness training. So that's what we used. And we ran this study. Before I get to the study design, just to note, and I think I mentioned this earlier, there is this very strong input into the hippocampus from the amygdala, the HPA axis, your stress response system, and the hippocampus actually is able to exert feedback inhibitory control over that pathway. It's also thought to exert a sort of contextual gating over pathways involved in motivated behavior, and it itself is modulated by input from prefrontal cortex. So cognitive control could exert its effects on the hippocampus, and the hippocampus could potentially shut down a stress response or magnify it. And it's noteworthy that when we add exercise into the loop, it has a big effect on hippocampal neurogenesis, well established in rodents, indirectly established in humans, that it upregulates neurogenesis, which actually has a protective effect against developing depression, so it impacts mood. I would call this via a more bottom-up route. It doesn't require cognitive control, basically. On the other hand, mindfulness training, or neurofeedback in our case, allows you to gain cognitive control over your brain state and potentially your mood state. So you're trying to figure out how to control your thoughts to go into this calm, relaxed state, which could potentially, sort of through top-down means, regulate the HPA axis. So we predicted that this combination would be even more potent than either one alone, acting via multiple pathways in regulating mood and in regulating those brain networks. So people with high depression levels tend to have too much default mode activation. The ruminating is, as I'll get to later, is associated with default mode activity. And people who have a highly mindful state are sort of in the opposite, and they have more alpha, more calm state, more balanced frontal activity. So that led to this massive study that actually I was chatting with John Iverson just before this, and I said, don't ever run a study like this. It nearly broke some of my students in my lab. But anyways, we did it. We put people pseudo-randomly into either a pure running group that ran twice a week for 30 minutes for 8 weeks, or neurofeedback with the Muse headband, similar spacing, or a combination, or nothing. That's a couch potato, in case you can't tell. It looks a bit more like a peanut, but it's a couch potato. We recruited people who were low active, so they exercised less than 1 hour a week total, and had no experience with meditation. And so there are many things that are interesting about the data, but I'm just going to focus in on the EEG. So now we again applied this alternating memory task setup while collecting EEG. We had people perform randomly interleave blocks of autobiographical memory and working memory. And we did that before and after the intervention. The VO2 max is a measure of cardio fitness. That was a manipulation check. And of course, the cognitive and mood batteries were also of tremendous interest, but I'm not going to say too much about them. We used the same EEG pipeline that I just described in the previous study. Well, I didn't really describe it. And just the classifier pipeline that Saurabh developed. And what comes out of it is these three numbers, the probability that the person is activating each of the three networks: default mode, central executive, and salience network. And then on top of that, Saurabh computed a quantity known as pairwise cross entropy, between the SN-CN cross-entropy or the SN-DMN. And when you have low cross-entropy, just intuitively what that means is that either signal alone is hard to predict, but in combination, they become easier to predict. You can predict one from the other, which implies low cross-entropy means high synchrony. So it's a nice measure indirectly of networks being in sync, co-activating, basically. Okay, so just one bit of other data, which I think is important, is that people naturally show a rise in stress levels from the start to the end of a semester. We've seen this across many studies, but those who were in the running group showed much less of a rise than the control. So that was nice. And they also showed improved VO2 max scores. I just want to focus, this is a busy graph, but I just want to focus on a couple of things here. One, in what I'm calling the responders. So we defined responders to exercise as those who had an increase in VO2 max, and responders to neurofeedback as those who were able to increase the amount of time they spent in the calm state, according to the Interaxon Muse headband. Okay, so responders from pre to post, those in the combined condition showed a very nice decrease in pairwise cross-entropy between the central executive and salience network when they're doing the working memory trials. On the other hand, those who were non-responders to the neurofeedback, interestingly, whether the neurofeedback alone or the combined group, showed a greater task inappropriate synchrony. So they're doing too much default mode network, salience network synchrony when they should be in working memory trials. So they're getting stuck in the wrong state, interestingly. And in fact, their pre-intervention levels of incorrect network synchrony, activating the default mode when they should be in the executive network node, predicted whether they would be a responder to the neurofeedback with about 80% accuracy. So you could actually use that. Not everybody does well at learning neurofeedback, and it's always been a bit of a mystery why that is. And we still don't know why, but maybe that's a bit of a clue. So that was also interesting. So what we've seen is that this combined condition seems to improve the synchrony between brain networks, and that these EEG feature classifiers are quite a useful tool at identifying which network is being active. Okay, so my last two topics for this talk, and we're doing well with time, I think I'm going to get through them all, are rumination and trauma memory. So rumination is Selena's PhD topic. And this is the title of a paper she has in preparation. She's published other papers, but this is the one I'm going to focus on for today. And it does link nicely in with our discussion around the default mode network and control networks, cognitive control, and so on. And just briefly, depressive rumination involves repetitive dwelling on negative thoughts, feelings, and past events. Of course, you can also ruminate on positive things. Rumination in general is thought to have two components, a reflective component that could be more adaptive problem-solving mode, and a brooding component that is not beneficial and is in fact linked to depression and suicide. And so that is what we're really focusing on, is the brooding aspect of rumination here in this talk. Okay? And one of Selena's very interesting predictions she's come up with is that this ruminative brooding might cause a failure to disengage the default mode network, especially these regions that are strongly implicated in rumination, posterior parietal cortex and posterior cingulate, which interestingly are also part of the perspective switching network. Again, remember those names coming up when we talk about trauma memory. But here it's very interesting that they come up in rumination. Now, I don't think that what people are doing with those regions is perspective transformations when they're brooding on negative thoughts. It's probably more likely they're just cycling through whatever. It might be more abstract things people are thinking about. You know, why am I a total failure in life? No, 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 no. That sort of thing. And it might be that it actually translates into a sort of pathological attractor state. That's where Selena's going with her research now, but we don't have those results yet, so I won't talk about that. So what we did here was look at measures such as, well, sorry, this is more background. People in depressive rumination show strong coupling between the amygdala and the hippocampus. That makes some sense. It's the emotional stuff. And also engagement, as I said, of the posterior cingulate and parietal cortex. So this is basically Selena's hypothesis that I just stated. And this slide should look vaguely familiar. We keep using that same task setup in the scanner. This time, though, we've added a string of rumination trials. So as before, prior to going into the EEG testing room, we had people generate a set of autobiographical memory narratives, but also a set of narratives of things they ruminate on. We did exclude people who have diagnosis of major depression, PTSD, and so on. So we're not dealing with a clinical sample. These are healthy undergrads, but they will vary naturally in their levels of rumination. And we did measure things like their trait and state rumination. And we expected that when they're engaged in a block of rumination trials, so these would be keywords about a particular thing they tend to ruminate on: uncertainties, future, stress. And then again, they're given a 4 alternative forced choice every so often to make sure they're on task. Which of your favorite ruminating narratives were you just ruminating on? And they would have to pick one. This is the cake one, so that's a neutral autobiographical memory in that block. And then again, we had a 2-back task. So they see a string of words. In every case, they're looking at a string of words, and then they're making a 4 alternative forced choice. But in this case, it's just which word did you see 2-back? That's important because in fMRI, you want to control for things like the stimulus complexity. So they're always just reading a string of words. And the response demands, they're always making a 4 alternative forced choice. And beyond that, they're thinking different things. And that's what we really want to image. What are they doing when they're doing these different tasks? So Selena took a different approach to analyzing the EEG data from what Saurabh did. So she actually used a beamformer to map from electrode coordinates into brain source coordinates. And she used several pre-selected brain sources, such as parahippocampal cortex. Hippocampus is a bit too small to hone in on from EEG, and other regions. Posterior parietal cortex and posterior cingulate being important ones for rumination. And then she filtered for gamma and beta power across the time series because these are thought to be correlative of cognitive processing. Okay, so this is a busy slide, but I just want to highlight a couple of things. So whether you're engaged in rumination, these are transitions. When you've been cued from rumination trials that you're now going into working memory trials, and down here you've been cued from having been just doing autobiographical memory trials, transitioning into working memory trials. So if you've been cued, if you have been ruminating, you might think there's going to be more carryover. It'll be harder for you to switch out of that mode. You'll have more sustained activation in that rumination circuit. And you might see that in particularly, say, the posterior parietal and posterior cingulate areas. And indeed, it does look that way. So this is the difference in power from pre-cue to post cue that there's a working memory trial coming up. So positive means you had more power in, say, the parahippocampal gyrus before the cue than after. Negative means you might have too much of that activity in parietal and posterior cingulate hanging around after you've been cued. You should be done with that default mode business and moving into your executive network and fully engaged in the working memory task. But if you're a high ruminator, that might be difficult. Now, these people are a whole range of high and low ruminators, but this is highly variable. With the autobiographical memory to working memory transition, there's also a lot of variability. But one thing that might explain that is now when we break this down by people's rumination scores. So what Selena did then was look at the correlation between their brooding score and this difference in power. In the parietal cortex is the filled-in dots, and the posterior cingulate is the X's. And you can see there is a weak but nice correlation. This over here is in beta power, this is in gamma power. Again, both are indicative of cognitive engagement. So more beta power in pre minus post, more beta power would suggest you're activating the parietal region too much. Have I got this the right way around? Yes, thank you. This is the least comfortable work for me because I only saw it like yesterday. So forgive me if I'm a bit, I'm looking to Selena for confirmation here. Yeah, so anyways, there's a nice correlation here with the brooding to the 0.34 between the brooding score and this power difference, which makes sense of that high variability. So those who were really high on brooding would have had more trouble disengaging. Those who were lower on brooding were probably okay at switching off that those default mode network structures and going into the executive network, presumably. And just, I didn't put a conclusion after that, but the next step here would be to look for evidence that there is actually some kind of attractor state or maybe a chaotic state where you're activating these regions too much and not getting out of that state when you should be. So those are the kinds of things Selena wants to do for the very last thing of her PhD. Okay, so we've talked about kind of healthy memory, memory in healthy individuals, memory in those who ruminate, and now we're going to talk about traumatic memories. And this is Mohammed's work. Mohammed finished his PhD in September, and he is still working with me, and he has a postdoc lined up in the coming months. So we are still doing some analyses, but I'm going to tell you about a couple of his results that are quite interesting. What Mohammed did was actually to access fMRI data that had been previously collected. In the first case, this is from a publicly available database from Vietnam War vets, and in another case, it was from PTSD patients. But in both cases, the interest is around post-traumatic stress disorder. Now, trauma memories are interesting, and it's debated whether are they an extreme form of quote-unquote normal episodic memories or highly emotional episodic memories, or are they a very different kettle of fish? Is it apples and oranges? And you can partly answer that by looking at what brain regions are engaged when you recall those memories, or even in the resting state. In the first study I'll talk about, it was collected during rest from the Vietnam War vets. In the second study, memories were collected while people with PTSD were in the scanner being cued to their trauma memory, which is something that's not easily achievable. Okay, so people who have PTSD, one of the hallmark symptoms is the vivid re-experiencing with all the emotional content, a flashback memory although they can be very fragmented and not necessarily like a well-grounded, well-contextualized episodic memory. People can be, like they'll think that they're actually reliving as opposed to knowing that they're revisiting a memory from the past. On the other hand, there is this interesting subset of those with PTSD who have what's called the dissociative subtype, where rather than having this highly heightened emotional response, they're just the opposite. Everything is massively suppressed, so they're numbed out, deadened looking, and that's presumably their strategy to dealing with overwhelming emotions of thinking back to a traumatic event. I won't talk about this in the first study, but I'll come back to it in the second study. All right, so the hippocampus. You might think, given that these are event memories, that the hippocampus would be really important for recalling trauma memories, not only for those who don't have PTSD, healthy individuals, but for those with PTSD. However, there is a prominent theory of PTSD, the dual representation theory, that posits that there's very much impaired hippocampal function, and that the reason these memories are so fragmented and not like normal autobiographical memories is that the hippocampus is not doing its job of setting the context, binding everything together, allowing you to know that you're recalling a past event. So that line of thinking would suggest, no, the hippocampus must be less involved in recalling trauma memories and PTSD. However, studies that have looked at functional connectivity in the hippocampus, which is essentially a measure of correlations in activations, if you're not engaging the hippocampus when you're recalling these memories, it should show this low functional connectivity. It's just not engaging with the usual network of regions, the default mode network, say. The studies have been mixed, and one thing that's been overlooked in most of these studies is that actually the hippocampus is functionally distinguished along the anterior-posterior axis in a way that mirrors in rodents the dorsal-ventral distinction, with the dorsal in rodents being where all those place cells are, the ventral being engaged in anxiety and fear behaviors and so on. So the emotional stuff is in the rodent, in the ventral. In mammalian hippocampus, the same thing is seen along the anterior-posterior. So anterior hippocampus is more associated with the emotional, fear, anxiety, etc. Posterior hippocampus is more associated with spatial representations and spatiotemporal context. So maybe if we looked at those two areas separately, we might predict that the anterior hippocampus would be more involved in trauma memories, in PTSD, and the posterior may be less involved. And in fact, this is Mohammed's model of the postulated changes between anterior and posterior hippocampus, with anterior in PTSD, the dotted lines, being more strongly driven by emotional cues, and the posterior being less connected to the emotional side. So that would predict you'd see less posterior involvement in PTSD, in recalling trauma memories, but more anterior hippocampus. So in this first study, Mohammed acquired this freely available data set from war vets, some of whom had PTSD and some of whom didn't. We excluded people who had head injuries, which was many of them because they were war veterans. And the main thing of interest was the PTSD distinction. And so when the whole hippocampus was seeded, like previous studies found, there wasn't a big difference in functional connectivity. However, when the anterior versus posterior hippocampi were seeded, and Mohammed looked at functional connectivity, there were a lot of regions. This is anterior hippocampus over here. I know you can't read those little labels, but you can see many lines of increased connectivity in the anterior hippocampus. And in terms of graph theoretic measures, the anterior hippocampus took on a more hub-like role, a more central role. Presumably, that would carry over from resting state to memory retrieval as well. So the next study I'm going to tell you about actually acquired data from Ruth Lanius's group, where they had people in the scanner either recalling a neutral memory or a series of morally injurious memories, which is a type of trauma when someone's experienced a moral injury. So these are considered to be traumatic memories. They're very common in PTSD. So you can think of it as a subset of trauma memories that are commonly seen in people with PTSD. And these are people with, they're either controls, they have PTSD, or they have the dissociative subtype. And furthermore, these memory recall blocks were divided into early, middle, and late time points to see if there was a difference. Remember when I described Sorab's results with the EEG and recalling autobiographical memories, and we saw an earlier rise in the default mode network, which made sense, you're recalling this autobiographical memory, and then a later rise in the central executive network. As the memory maintenance part of the task becomes predominant, you go more into the working memory mode. So that was why we thought it was important to look at these different phases of memory. And this is a busy graph, but I just want to highlight a couple of things. So first, let's look at those with PTSD, the red lines. I hope nobody's colorblind and can't see the red versus the others, but I'll also point out which I'm talking about. So those with PTSD are on top in terms of activating the salience network, what's been called in the literature the innate alarm system, which includes some subcortical areas, such as the periaqueductal gray. I didn't define those, but it is expected that you'd see that. And so they're activating these and the anterior hippocampus most strongly. So that's those with PTSD. And in terms of cognitive control, they're really low. They're bottoming out here. Ventromedial prefrontal cortex and dorsolateral prefrontal cortex, those with PTSD are not under cognitive control, but they're activating subcortical areas associated with emotion and the emotional side of the hippocampus. What about those with the dissociative subtype? They're the ones that are really shut down. They're at the bottom in terms of activating the salience network, the innate alarm system, at least until that very last time point they're able to suppress. But they're at the top of the class when it comes to cognitive control. Ventromedial prefrontal cortex and dorsolateral prefrontal cortex are all pretty high. And interestingly, they're also activating this perspective switching circuitry, which we really think they're engaging because they are performing a perspective transformation. It's well known that people who are high in dissociation tend to take a more third-person perspective when they're recalling things, which is a way of dampening the emotional content. So it's a way of getting emotional distance. So it's very interesting that they would be engaging that circuit more, as well as the cognitive control, which goes along with that. There are also other subcortical areas that come in that are interesting with these traumatic memories, the vestibular and some motor areas in the cerebellum. In terms of the healthy individuals, interestingly, you could almost say they're in between those with PTSD and those with PTSD plus dissociation on many of the measures, not all. They're not as high on activating the anterior hippocampus, but they're effectively engaging their posterior hippocampus. That's the more usual suspect you would expect in recalling an autobiographical memory. And so even though it's traumatic, it's a morally injurious memory, they don't have PTSD. So they're able to engage that posterior hippocampus and presumably have some emotional distance. They have some cognitive control happening near the beginning, but it's not as high as and maintained throughout as the ones who have high dissociation. All right, and then less anterior hippocampus, more posterior in the healthy individuals. So it's a really interesting pattern of data contrasting these three groups. There's a lot more to be done to really be able to say, oh, trauma memory is the same no matter whether you're in a clinical group or a healthy group. You need to look at fine differences in what the subcortical regions are doing. And there's a really under-recognized role for areas like parts of the cerebellum, the emotional/cognitive parts, in memory recall, in emotional and traumatic memories. And this so-called innate alarm system that Ruth Lanius's group has actually published many papers on, that is highly engaged in PTSD and highly suppressed in the dissociative subtype, which we're seeing here too. So that's reassuring. Yeah, so just to conclude there, trauma memories seem to engage the anterior hippocampus, the innate alarm system, and the salience network, especially in those with PTSD, whereas we see the opposite in those with the dissociative subtype. They're not so much engaging the anterior hippocampus and the innate alarm system, but they're engaging cognitive control networks and potentially this perspective switching network, which might be allowing them to shift the viewpoint and maintain that emotional distancing. Yeah. So that brings me to the end of the main results. Just to sum it up, the default mode network, it's not just the brain's idling state or task neutral state. It's engaged in a variety of tasks, including episodic and spatial memory, rumination, and trauma memory and PTSD. When there's dissociation, we in addition see the engagement of cognitive control and perspective switching networks that seem to dampen the emotional side of the memories. And in rumination with the brooding subtype, it could be that the person is getting stuck in some kind of pathological attractor state or chaotic system that's spinning around, and they're having difficulty disengaging that. So there's a lot of interesting work to be done there as well. All right, so there's actually an awful lot of undergrads who contributed to this work, and it would take me 10 slides to list them all, so I didn't list any, which I apologize if any of you are in the room. But these are the graduate students who were the leads on the work, and some of our main collaborators and funding sources. Interaxon generously donated a whole bunch of their Muse headbands, so we were very fortunate to have that as well. And lastly, I'm retired now. John did mention that. That's what I've been doing.
So we do have time for some questions. Sigal in the back.
Hi, Steve. Thanks for a really interesting talk, and welcome back.
Thank you. I miss you too.
So I have a question about sex differences.
Oh, yeah.
And there are well-known sex differences in how frequently we see post-traumatic stress disorders, or whatever they're called. And the dissociated subtype specifically, what do you think your results might tell us about those sex differences, or can they?
Oh.
I know you didn't
I should know this, but I know the sex differences very well in the depression literature, but not so much in the PTSD literature.
It's supposedly twice as common, maybe even more
In females, including the dissociative subtype?
No.
Not including.
They're not very common.
Oh, interesting. Oh, wow. Yeah, that's a great, obviously I haven't thought about this because I didn't even know about it. But in depression, there are many established sex differences in brains on average, with one being that the average male brain is more strongly lateralized, less interconnected across the hemispheres. Our own Sandra Whittleson has done some of that groundbreaking work here in the neurosciences. And so you might think that more hemispheric crosstalk would protect you, because depression is often thought as being too much stuck in a right hemisphere mode of thinking. And you might have thought the cross connections would make it easier to transition between the two. So I don't have an explanation. I find it very puzzling. And then in terms of PTSD, I mean, well, we know that sexual abuse is more common for the experience for women. But I mean, first responders, I think the PTSD rates are high for both men and women, and war vets. So you might need to break it down by trauma type. I'm not sure. And in terms of the dissociative response, that's interesting too, because I think, Selena, from your looking at the rumination literature, it was more right hemisphere in rumination, which fits with the right hemisphere predominating in depression. When people use TMS to try to re-engage part of the brain to treat depression, it's typically the left frontal region where it's applied to try to get more left hemisphere activation going. I'm giving you random information, talking around your question, because I don't know the answer. I'm just giving a few clues to how I'd even begin to think about it. Does anyone else have any thoughts about that? Such a good question. Yeah, no, we'll definitely be thinking about that. Thank you. Yeah. Hi. Thanks.
In the dissociative subtype, might the perspective switching come from not a conscious effort to distance yourself from the emotions of the traumatic event, but rather from the inability to access the somatic emotional aspects?
Oh, that's interesting. It's kind of a chicken and egg question.
Yeah, so instead it would come perhaps more from your only conscious access point to the memory is through more procedural kind of third person events rather than the actual emotional contact.
It could be. It could be. I mean, I know it is common in sexual abuse when people can't tolerate the bodily sensations, and they tend to become disconnected from those anyways. Even in flashback memories, they tend to disengage the bodily sensations. And actually, you can see that in Muhammad's connectivity findings that I showed, that the one blue line where there was decreased connectivity, that's to the supramarginal gyrus. So that sort of hints at a sort of decreased connectivity with areas involved in bodily sensations. Yeah, so that's very interesting. But which comes first, that or the dissociation and the switch in perspective, is a great question. Yeah, and that would be tricky to disentangle. Very good question. Thanks. Meyer.
Thanks, Sue, for the talk. So at a high level, your presentation makes me think of, like, clearly there are emotional paths and then prefrontal paths to kind of controlling memory. And I suspect that there are lots of individual differences in whether you remember things much more emotionally or much more objectively, from a cognitive perspective. So in your data about these different networks and to the extent that they're activated or how quickly one switches, like, do you see any trends that might give us insights into these, like, the differences in how people do reflect, or how they, to what extent these two networks are impacting how people remember?
That's interesting. I mean, what would be nice to try to answer that would be to try to do a controlled study where people recall events of varying degrees of emotionality and emotional valence, and then track the time course of the memory activations. And then there might be individual differences overlaid on top of that, which you're getting at, which could well be that some people, it doesn't matter if it's a very emotional memory, they can just easily reactivate it and recall it, and not so much activate the emotional structures, and some people might really readily do so.
relative difference in the networks is seen consistently in every individual?
Well, I don't know. We probably don't have enough data to really do individual differences in that way. But if you had a data set of a few hundred people, you could. These are hard studies to do where you get someone sitting in the EEG lab for an hour plus half an hour setup time plus, half an hour of hair washing afterwards, doing your tasks in this electrode cap, getting a headache. So it's hard to get a very large sample size with those studies. Yeah, but it would be ideal. Thank you.
Time for one more question? Okay, well, let's thank Sue.
Oh, is there one more?
Sorry. Were there any differences between the dissociative subtype and control in the neutral
In neutral memories. I'm going to shunt that to Mohammed.
Yeah, the data is not shown here, but yeah, the healthy controls actually in the neutral memory, they activate at the perspective switching like more than like the perspective.
A reversal.
Yeah,
That's
that's the
interesting.
one result I remember. I have to look at my results, but yeah.
Huh. Which could also be, it might be perspective switching, or it might be memory maintenance or something like that. Those same structures come up in many tasks, so we have to try not to overinterpret our data, even though we really want to. Yeah.
Okay, well, thanks so much, Sue.
Thank you. Thanks, everyone.